Okay, vision part 3. Oh my gosh, will it ever end? Vision is a little complicated. I talked about that earlier, but you need to know that the difference between like the sensation and the perception. So the sensation of light and then how your brain perceives it. So we've talked about the biology of the eye. We've looked at the different parts of the brain that are associated with your optic nerve and vision. And now we're going to actually focus in on light, so that the physics part of light. Okay, so we're going to look at color in this and then feature detections as well. Make sure that you are drawing things that you see as well as writing them down. So you'll need to know like the properties of light and be able to identify what they are. So vision, you need to know this is a big vocab word, transduction. Okay, so transduction is the actual process of converting one form of energy to another. So I'm converting vision or light energy into chemical energy so that it makes sense in my brain, right? Because we talked about neural transmission is really those chemicals that are released when my axons or my neurons fire. So transduction, converting one form of energy or light into another. So in sensation, when we have all these energies that we are sensing from our outside world, that's talking about transforming the stimulus, so sound, so the air vibrating, vision, light, sense, weight, pressure, things like that, into neural impulses, which are going to be chemical. Okay, so for vision, we're going to talk about transduction in the form of wavelengths. So you can write down wavelength, but we're going to come back and look at that. But really wavelength, we're only attuned, so our eyes are only attuned to see certain wavelengths of light. All right. So the three things we're going to talk about, so wavelength and then hue and intensity. Hue is going to be our color. All right, so you can think hue means color, and color is determined by wavelength. And then our intensity of light is going to look like how intense it is. Is it really bright? Is it really vivid? Is it loud? So that's going to be determined by amplitude. So light can be both described as a particle and a wave because it travels in waves, but it kind of acts like a particle. A wavelength, you're going to draw this in just a second, is the distance from one peak to another. You might know this from physics. And you might know that visible light, the light that we're able to see, only ranges from 400 to 700. And there's this whole other spectrum of light that we are not able to see. We might be able to feel it, like infrared light is heat. UV rays might burn us, but we're not able to see them. And we actually determine color of light related to its wavelength. So here are my physical properties. You need to sketch these out next to what they are. So from peak to peak is going to be a wavelength, all right? So my really short wavelengths are associated with bluish colors. All right, so you can think like UV rays or violet get really, really close together. Those are going to be my bluish wavelengths. Versus a longer wavelength are associated with redder colors. These longer wavelengths go on the infrared side of the electromagnetic spectrum, like associated with heat. So you can think that the reds are closer to the infrared, the blues are closer to the ultraviolets. All right, so that's my wavelength. How tall the wavelength or it is from bottom to peak, this is called my amplitude. So bright colors have a higher amplitude, and then dim colors are going to have a smaller amplitude. All right, so here's my spectrum again. So if I took white light and put it through a prism, it would actually be broken up into these different wavelengths. This is just what I explained to you. So I have my longer wavelengths over here that are going to infrared, my higher wavelengths here that are closer to blue, and then everywhere in between. So the mnemonic device for this is the Roy G. Biv. So you've got red, orange, yellow, green, blue, violet, and that's looking at energy going from longer wavelength to shorter wavelengths. Okay, now our rod and cones are able to look or receive different types of light. So remember that cones are specialized for color, the rods are specialized for black and white. So this says the rod and cone behavior is illustrated by these two fire engines. Note that in daylight or bright artificial light, both engines are visible. All right, so I can see right now this red and blue. And then in class, we're going to actually, we're going to go to a dim light or a dim lit area and see what that changes when we get into darker areas, if we can still see those colors. All right, so color vision is talking about our visual systems, how we interpret color in the terms of wavelength. Okay, so remember, rods and are colorblind, so they're just seeing black and white, and then our cones are attuned to those different wavelengths. So when that difference occurs, or the difference in colors occurs, because we only have one type of rod, but 3 different types of cones. So the reason we're able to see different colors is because we have 3 different types of cones. Blind spot. Oh, I just put this here for review. So the blind spot, this is when we talked about those are where our optic nerves leave the back of the retina, and it creates this blind spot in our visual field. We will actually look at your blind spot in class. Okay, so color blindness we'll come back to in a little bit, but let's talk about feature detection. So feature detection is really when, so we have all this light stimulus coming in and it's converted to chemical energy with our neurons, but our nerve cells being able to respond to different features such as shape, angle, and movement. All right, so our stimulus might look like this, and what that looks like in our neurons might look something like this. Okay, so our cell's response doesn't always exactly look like our stimulus. And I want you to now kind of start focusing on sensation versus perception. So what is actually sensing coming in and how my brain is perceiving it. All right, so let's look at this next picture that has shapes, angles, but no movement. So shapes and angles. So this is called the nectar cube. And really all it is lines. Okay, so I actually have lines here. This is a 2D object on the paper, but when you look at it, you can actually see the 3 dimensions of the cube, so the nectar cube. So think about this now with sensation and perception. So really the sensation coming in is these lines, but we perceive it to be 3D. I can actually see like the cube going back and making a 3-dimensional space, even though there's only 2D lines. Okay, so let's start focus, like again, like this is what you're sensing, but that's not what your brain is perceiving. All right, here's some other optical illusions. So these are like little Pac-Man shapes. There's no line here, right? But our brain perceives a triangle. It kind of fills in the gaps where we can actually see a triangle, hopefully you can, that goes right here. All right? Now that triangle doesn't exist if I take it away, but my brain still perceives it. So that's an illusory contour. It means that that contour does not exist, but my brain still sees it. So those are our shapes, and now let's talk about movement. Okay, so our brain is also involved in like determining whether or not something's moving. And this is done with a process called parallel processing. So parallel, think like it's right next to each other. So 2 things are happening at the same time. All right, this is really important for movement. So we need to be able to constantly be taking things in and processing at the same time. So there's 4 different things we're going to talk about with parallel processing. First of all, color. All right, so color vision will be separate. Second of all, motion. So when things are moving, right? So moving from one place to another, how our brain perceives the stimulus as a perception. Form. Okay, so we actually have something called form constant, so where our brain is able to keep a certain form for objects. And then depth, and that's looking at the 3D-ness of an object, like thinking at that nectar cube. All right, so here's how our brain visually processes. This is going to be a step by step, so make sure you write them down. So first of all, we have the scene. In this case, we have the divers, all right? The scene before was the bird, so whatever you're looking at. So from the scene, it goes to our retinal processing. Retinal thinking your retina, so just like your eye. So it's going from my rods and my cones to my bipolar cells to my ganglion cells that leave the eye. Oops, sorry, I went the wrong way. And then that's where it goes to our feature detection areas of our brain. So our brain detects, is it their edges, is their lines, is their angles, is their movement. And from there, we have parallel processing that starts to occur. So the brain cell, look at that, teams processes where it's combining information coming in about color, movement, form, and depth, and that goes on to recognition. So all these things are happening at once for our brain to interpret and construct images based on our stored information. So I know that this is a diver. I know that they're probably in the middle of the air, and the next thing I can predict has happened is they're going to like straighten out their bodies and dive in. All right, and then it goes back to the scene. So see how it's kind of a continual process? So I detect features, parallel processing. My brain interprets it to something, and then it goes back to the scene, and that process keeps going on.